it's working now. So.
Right. So, hi everyone. No, okay, not yet.
We're getting there.
Hi everyone, welcome back to our early career immunology seminar series. Today I have the pleasure to introduce Ballesteros, who is going to talk about the functional organization of the innate immune system. He studied biology and obtained his PhD in pharmacology in 2012 at the Universidad Complutense de Madrid, where he characterized how the presence of immune diversity critically determines the outcome of stroke. Later, during his first postdoc in Memorial Sloan Kettering Cancer Center, he characterized the genetic programs that emerged during development and that instruct macrophage heterogeneity in tissues. For the past 5 years, he has worked as a postdoctoral researcher at the Centro Nacional de Investigaciones Cardiovasculares, where he has led studies exploring the diversity of neutrophils in steady state and discovered that tissues co-opt neutrophil fates to induce transcriptional programs. He's currently a Ramon y Cajal Fellow at CNAC and continue to explore the architecture of the innate immune system to generate novel therapeutic strategies to fight disease and to develop new diagnostic or prognostic tools. Thank you very much, Ivan, for accepting our invitation to present today, and we are looking forward to hearing from you.
Thank you very much for your introduction and for the invitation to present here in these seminars. I'm going to share my screen and put it in presentation mode. Okay. So I'm going to talk about this organization of the innate immune system. And basically, the cells of the innate immune system are very heterogeneous, and we are trying to identify ways to generate models to understand this heterogeneity. And this is important because this heterogeneity is highly associated to disease outcome. For example, recent examples in COVID infection we know that different types of monocytes or neutrophils associate with the severity in COVID patients and also in cancer. It has been known for years that different phenotypes of macrophages associate to different outcome in cancer. I did my studies, PhD studies in the field of stroke. When there is a stroke in the brain, there is an area that is irreversible, cannot be rescued. But surrounding this area, there is a penumbra, an area with a low blood flow that can be rescued. And we found that modulating the inflammatory response, we can protect the brain and rescue some of the tissue that otherwise it could be dying. And we associated that different types of innate immune cells associate with this response to a stroke. Basically, that's why it's important, this heterogeneity. I focused on the neuroinflammation. But indeed, to understand how this is happening, we need to better study the biology of the cells of the innate immune system. And I focused on the study of macrophages and neutrophil biology. And today I'm going to focus on basically on the neutrophil biology part, but I will talk a little bit on macrophages because I like to compare both different cell types together. One of the first differences that you can identify between tissue resident macrophages of most populations of tissue-resident macrophages and neutrophils is their origin. Tissue-resident macrophages originate from early progenitors in the yolk sac, in the embryo, and in contrast, neutrophils arise from hematopoietic stem cells that continue self-renew, give rise to maturing neutrophils, and these neutrophils will go into circulation. So regarding to the biology of these tissue-resident macrophages, one, they originate in the yolk sac very early in development at early E8.5 in the mouse, and they migrate to the fetal liver, and they colonize tissues and give rise to the tissue-resident macrophages that we found, for example, in the case of the brain, to the microglia. So I basically explore during my postdoc with Dr. Geisman the initial time points of the development of these macrophages. And here you have a single cell analysis of leukocytes from an E10 embryo. So you can identify different populations, the progenitors, a transitioning population, and a more mature macrophage population that is characterized already at this time of development by the expression of markers like scavenger receptors, mannose receptor, Fc receptors, or complements. So very early in development, you already have this macrophage signature. And if you follow these macrophages across development in different tissues, one of the things that you can find is that as early as the macrophage colonized its tissue, in this case, you can look here at the microglia, it already established its identity. This identity that we identify in tissues in the adults, that it's associated to specific functions and to specific phenotypes. So we now believe that these macrophages are indeed components of the tissue, minimal components of the tissue, together with the tissue-specific parenchymal cells, endothelial cells, stroma. They accomplish very specific tasks in a tissue, and this is possible thanks to their specification that, as I told you, take place very early in the embryo and very fast, and then is maintained across the lifetime. Evidences show that the environment is key for the acquisition of these specific functionality of tissue-resident macrophages, together with the transcriptional control, intrinsic transcriptional control in response to this environment. So we have here, we have here a comparison between a macrophage and a neutrophil, and we can find differences, like, for example, in terms of lifespans. These macrophages, these tissue-resident macrophages live longer times. In contrast, the neutrophils are produced daily, and they have very short lifespan. When we talk about the proliferation, we know that in early times of development, these tissue-resident macrophages proliferate a lot, but then they are quiescent in a steady state, or the proliferation rate is very low. In contrast, with the neutrophils, we have a production of neutrophils in the bone marrow that happens every day. We produce massive numbers of neutrophils. And regarding to the specification that I told you, there was a big question for us. We wanted to know if these neutrophils are also able to specify, and if they specify, if they were able to accomplish tissue-specific tasks as the macrophages did. So when I joined Andres Hidalgo lab, we knew that in healthy tissues, you can already identify neutrophils. These neutrophils are present in these tissues in different numbers. For example, you have a lot of neutrophils in steady state in the spleen or in the lung, but very few neutrophils are associated to tissues like the intestine or the skin. And when you look at the localization of these neutrophils in the tissues, in the steady state condition, you have different patterns of localization. If you look at this micrograph here, this is the colon submucosa. This is an area of the colon submucosa surrounding the isolate lymphoid follicles, and these areas are full of neutrophils, and these neutrophils, as you can see, they locate extravascularly. In the case of the lung, we can see that most or all the neutrophils are associated to the vessels, to the parenchyma, sorry, to the small capillaries of the lung. Of course, we did this by perfusing tissues and taking out the blood, and some people can argue that this is contamination, passive contamination of the blood, the remaining blood that stay there. We have some evidences that this is a active process, and one of these evidences comes from the circadian biology of the neutrophils. They are released to the circulation, and they follow these circadian patterns in which you have high number of neutrophils early in the morning, and they reach a plateau in their numbers in the early evening. So if you compare this to what we see with lung neutrophils, you can see that these curves are antiphasic. So they suggest that indeed this accumulation of neutrophils in the capillaries of the lung is not a passive event. It's kind of an active event. So the next question that we wanted to answer is how long do these neutrophils that we find in tissues live? Because the idea is that once the neutrophil is in a tissue, is phagocytosed by resident macrophages of tissue phagocytes. So we generated this model in which by injection of tamoxifen, you can induce expression of tomato in neutrophils. So we injected tamoxifen to a mouse, and we analyzed the number of neutrophils, red neutrophils in different days after tamoxifen injection and in different tissues. So with this information, we were able to build a mathematical model, a curve of tomato decay, in which we could analyze the half-life of the neutrophils in the different compartments that we explore. And as you can see, they are not immediately eliminated from the tissue. They can stay there for around 10 hours in the case of the lung, or more than 18 hours in the case of the spleen. And if you look at the lifespan of these neutrophils, they are much longer. They can be there for more than 1 day. So this gave us an idea that the neutrophils stay in the tissue environment enough time to change. And that's what we explore. We explore how different neutrophils are depending on the tissue of origin. For that, we did different strategies. The first one was at the protein level. We perform mass cytometry, and these are leukocytes from different tissues. And if we look specifically to the macrophages, you can see that they nicely clusterize depending on the tissue of origin. But if we look at the neutrophils, we can see that they can also clusterize depending on the tissue of origin. This is at the protein level by mass cytometry, but we also identify tissue-specific transcriptional profiles by RNA sequencing in neutrophils from different tissues, as you can see here in this heat map. And this percentage of changes was very comparable to the one identified in macrophages. In addition, we also found that these changes in the phenotype in the neutrophils of different tissues was also associated to the DNA. This is attack sequencing experiments, and you can see that the neutrophils cluster depending on their different tissues where they come from, so showing us that there were also changes at the DNA level. So with all this information, we propose that neutrophils could also accomplish homeostatic functions in tissues that in some cases were specific from their tissue of residence. You have here the example of the blood and the bone marrow, where you can see chemotaxis and immune response are main functions of the neutrophils as expected and as it has been demonstrated for several years now. But if we look to other organs like the lung and the intestine, we start identifying some functions that were non-canonical for a neutrophil, like angiogenesis and neuronal development. One of the interesting things that we also identify, and this is the case of the lung, but we think it can also take place in other tissues, is that neutrophils located in specific areas where they could reprogram and acquire a tissue-specific function, like mirroring the biology of tissue-resident macrophages. In the case of the lung, these neutrophils located close to CXCL12 positive vessels, and these allow them to specify. And if you disrupt this axis, I'm not going to go into details, this is already published, but if you disrupt this axis, the neutrophils cannot acquire their angiogenic function in the lung. So basically, mirroring the biology of these two cell types of the innate immune system, we prove that the neutrophils can also specify in tissues, acquire tissue-specific task or tissue-specific functions. The main mystery here is, okay, so we have two different cell types, tissue macrophages and neutrophils. They seem to behave similarly. They seem to acquire a functional specification. We don't know exactly how effective is a neutrophil in performing tissue homeostatic tasks, but the macrophages, we know they do it very efficiently. Then this is for us a mystery. Why do we have two cell types very similar in terms of functionality and biology? Why this is so redundant? Why this is taking place at the same time? And we don't know exactly why, but we really want to understand better the biology of the neutrophils. And now, thanks to single cell omics, we are able to identify heterogeneity in neutrophils with unprecedented resolution, and others have already performed some modeling to explain the organization of the granulocytic compartment to better understand neutrophil biology. One of these models show that neutrophils organized across a developmental trajectory, a single developmental trajectory that they call neutro-time. So it's a maturing trajectory that indeed contains a core neutrophil transcriptional profile from the bone marrow to the circulation, and giving rise to very homogeneous populations of neutrophils. In the other in other studies, they also prove that this heterogeneity of neutrophils, but they identify that there were a specific neutrophil progenitors or populations in the bone marrow that were able to give rise to different population of neutrophils in the circulation. So, and in addition, the study I already told you that we perform on tissues give us another layer of heterogeneity, in which it seems that tissues are specifying neutrophils, are co-opting neutrophil fate sites and giving more heterogeneity. In the first model, in the nucleotide model, we can explain it like this continued maturation from the bone marrow towards the circulation. But when we add the other two models, the complexity increases because they are claiming for a peripheral reprogramming of neutrophil in any level or in the bone marrow or in the tissues. The main question is how this is happening, which are the mechanisms that perform this peripheral reprogramming of neutrophils. I'm going to talk about environment mainly today, but there are other options like pre-commitment of these neutrophil progenitors that may give rise to specific neutrophil populations. So we wanted to explore this more in detail, and specifically because we want to understand what happened after a stress. We have all these models, and then there is a stress, and several things can take place. We know there is a lot of heterogeneity. It has been shown in cancer, in infection, but the main question is how this is organized. So when there is a stress, there is a de novo compartment, new neutrophil subsets arises, or indeed, this is just an expansion of a neutrophil compartment that was already there, and there's no changes in the whole architecture of the granulocytic compartment. So we start exploring this, and because we know that the neutrophils are very heterogeneous, as I told you, and we know some of the programs that are associated to this heterogeneity, like maturation, circadian agent. But when we talk about the heterogeneity that we find in steady state or in disease, it is less clear how this is originated. So we start exploring this using single cell sequencing analysis. Here you have neutrophils from the bone marrow. And if you look, this is a UMAP clusterization, and you can find a group of cells that are in proliferation. You have a maturing track of neutrophils. We have more mature neutrophils here that do not seem to have any specific function, and some neutrophils that express class 2. Of course, we can take advantage of the heterogeneity that we find in the steady state and add more neutrophils from different tissues to this map and build a more complex map on neutrophil heterogeneity. And that's what we did. We add neutrophils from the blood, lung, spleen, liver, and gut. This is all a steady state. And you can see that we have here again the maturation track that we had previously when we studied neutrophils from the bone marrow. We have the proliferation subset, and we have the class 2 here, and we start seeing some potential functional areas in the map. Interferon, neutrophils associated with interferon responses, and neutrophils associated to angiogenesis and immune suppression. We can perform some trajectory analysis on these maps, and this is a slingshot showing us that we can identify two main trajectories in the map. One leads to interferon responses, the other leads to angiogenesis and immune suppression. And on top of this, if we look at this map, you can see that it's compartmentalized depending on the tissue of origin. You have here in the maturation tract, most of the bone marrow neutrophils. In the angiogenic and immune suppressive area, you have mostly lung and neutrophil and gut neutrophils. And for example, in the interferon tract, you have most of the spleen associated neutrophils. So if we explore this functionally, what we could expect according to this data is that the neutrophils from the lung are more angiogenic and more immune suppressive than, for example, the neutrophils from the spleen or the neutrophils from the bone marrow. So we explore this functionally. First of all, we check angiogenesis activity in neutrophils. To do this, we perform a matrigel assay in which you inject a matrigel subcutaneously, and then to these matrigels, you inject neutrophils coming from different tissues. In this case, I showed you a matrigel in which we injected tissues from the blood, neutrophils from the blood, and another matrigel in which we injected neutrophils coming from the lung. And you can see here, this is a staining for vessels, and you can see that in the matrigels where we injected neutrophils coming from the lung, we have more number of vessels. If we explore the perfusion in this matrigel using Doppler imaging, what you can see is that in the matrigels where we injected lung neutrophils, we had a higher perfusion. So indicating that indeed neutrophils from the lung are more pro-angiogenic than neutrophils from other tissues, as expected according to our single cell dataset prediction. And then we also check immune suppression, and to do that, we perform an analysis on the efficiency of T cells in killing cancer cells. So cancer cells express OVA, and these OT1 cells recognize OVA. And as you can see, this is crystal violet. This is a plate in which we have all the cancer cells here. When you co-incubate these cancer cells with T cells, you see that they efficiently kill these cancer cells. When we co-incubate these T cells with neutrophils from the blood, from the spleen, or from the bone marrow, what you can see is that they also can kill the cancer cell very efficiently. But interestingly, when you incubate these T cells with neutrophils from the lung, this killing is diminished. So showing us that indeed these lung neutrophils, they have an immune suppressive activity. So I want you to check on this graph here. This is a trajectory. This is trajectory number 2 that leads to pro-angiogenic and immune suppressive neutrophils. And you can see that there is a gap here. So indeed, all this trajectory analysis try to put everything together. But when there are gaps like this, what we think it means is that we are missing some of the heterogeneity of the neutrophil compartment. So indeed, what we wanted to do is to have the most representation on the heterogeneity of a neutrophil. And to increase this, we add more neutrophils in our data set, more neutrophils from steady state tissues, but we also add diseases like, for example, cancer, or neutrophils from different physiological conditions like pregnancy, or neutrophils from other tissues like placenta or the meninges. And doing this, we have another map of neutrophil heterogeneity. And if you look at this map, there are interesting things that we can identify. The first thing is that there are no more functional states emerging compared to the other map. You have the interferon area here, the angiogenic area here, the class 2 neutrophils integrates much better in the map. And the good thing is that we have very well separated cluster, much better separated clusters that define different functional states, but we don't have new clusters in this representation. So having this, we wanted to explore some of the biology of the neutrophils, taking this as a tool to do so. The first thing that we do is to check if mirroring macrophage biology, these states were associated with transcriptional factor activity. So I'm not going to go into much detail, but we identify that in the case of the lung, JUMB seems to be a transcription factor that control the specification of neutrophils. So what we did is to generate mice with neutrophils deficient in JUMB and perform single cell sequencing on neutrophils from these mice. Here I'm just showing you the lung neutrophils. These are lung neutrophils from wild type animals. These are lung neutrophils from that do not have JUMB. And we compare other tissues, but the differences are specifically in the lung. There's no differences in other tissues. And the differences that you can see is in this area here, this area that is associated to the angiogenic and immune suppressive cluster. If you look at the JUMB mutant neutrophils, you can see that they do not have this cluster. So suggesting us that indeed, when there's no JUMB, in steady state conditions, neutrophil cannot give specify toward this subset and or toward this functional state of angiogenesis and immune suppression. So we tested this functionally, and if you look at the graphs of immune suppression, you can see that in the case of neutrophils from the lung of JUMB deficient mice, you can see that the immunosuppressive activity of the neutrophils is reduced. And if you check the angiogenic capacity of the neutrophils coming from lungs from JUMB deficient mice, you can see that they cannot produce more vessels. So indeed, we identified that this JUMB activity was required for this immune suppressive and angiogenic function of the neutrophil. And another thing that we identify in this map is that these neutrophils do organize in a finite number of functional states. There are very few functional states, even if you add different conditions and different contexts. Here I'm showing you again this angiogenic and immune suppressive cluster, and you can see that this cluster is not only present in a steady state lung, but you can see this cluster of neutrophils. So this profile of neutrophils, for example, in the intestine, or in the placenta, or in the pancreatic tumor, neutrophils in the pancreatic tumors, they are mostly having this transcriptional signature, or after myocardial infarction, you can identify this angiogenic and immune suppressive cluster. So this indeed to us pointed to this idea of when there is a stress, the architecture of the granulocytic compartment is indeed maintained. So there's not the novel new phenotypes of neutrophils. Indeed, what it seems that is happening is that there is an expansion of already existent activation states or functional states of the neutrophil compartment. We believe that indeed environment is important for that. We think that in these conditions, there may be physical structures that instruct neutrophils to acquire this specific function. And I'm going to finish by showing you an example on neutrophils in the liver. These experiments we perform with Pau Sancho and Silvia Ariño, that are expert in liver biology. We perform a model of liver injury in which you can see that in the control liver, there are very few neutrophils. Most of them are intravascular, but when you perform a damage, you have a lot of neutrophils, and they locate in a specific areas of the liver. In this case, they locate very close to the biliary trees in association with the epithelium of the biliary trees. And we think that this is indeed similar to what we find in the lung. We think that the CXCR4, CXCL12 axis is important for this localization of the neutrophils in the liver. And I just want to show you, like transcriptionally, how the neutrophils from a steady state liver that is here change in comparisons to a liver where we have this kind of injury. You can see that neutrophils moves from this area of the map that we built towards the area of immune suppression and angiogenesis. And so it kind of suggested us that maybe they are going to a specific area of the liver where they can reprogram, as what we already saw in the lung. And indeed, if you remove CXCR4 from neutrophils, so they cannot sense CXCL12, the first thing that you see in a liver with this kind of injury is that the neutrophils, they do not longer associate to the vascular tree, to the biliary tree, sorry. They associate randomly in the biliary tree and in the parenchyma. And this, in the case of CXCR4 mutants, so neutrophils that cannot go there, what we find is that there is a parenchymal damage in the liver. These are biliary lakes, probably in response to biliary acids that are secreted into the parenchyma. So in this example, what I want to show you is that localization seems to be very important for the function of a neutrophil. And I'm just going to finish with this, and just to make a small recap of all I told you on our ideas of the granulocytic system. First of all, they are organized in a finite number of functional states, and we can find that there are convergent phenotypes across different biological conditions, very, very different biological conditions and physiological contexts. Of course, they are safe upon disease. We can reprogram them. At least we identify that transcription factors really control these functional states. And we believe that there are physical structures that are determining this reprogramming of the neutrophil. So with all this in mind, we think that we can start thinking about new strategies to reprogram these cells. Knowing this biology, we can, for example, target directly the stroma or the environment, or target different transcription factors to modulate the function of the neutrophil and to fight diseases. So this is basically everything I wanted to tell you. I would like to acknowledge everyone with I've been working for these years and institutions, and more especially to Andres Hidalgo, and that is a principal investigator of the lab, and we are working very close in defining this heterogeneity of the neutrophils together with Dr. Laiwan Eng, and also Daniela Cerezo and Andrea Rubio, that are two postdocs of the lab that are working very close with me in defining these functional states of neutrophils and trying to make sense of the architecture of the granulocytic compartment. And that's all. Thank you for listening, and thank you for coming.
Thank you, Ivan. That was really great. Very impressive amount of work. So we do have some questions in the chat already, so I'm gonna go through them. So we have a first question. Are there tissue-resident neutrophils in steady-state liver parenchyma?
We identify neutrophils in a steady-state liver in the vasculature, inside the vasculature, the vessels, but not in the parenchyma.
Mm-hmm. I think you showed actually some of that
Yeah.
in the later results. Yeah.
Mm.
The second question is, what exactly are class 2 neutrophils?
So that's interesting because we are trying to make sense of them. We don't know exactly what this means. We don't know if this is a response of neutrophils to damage, for example, that interferon signaling somehow make them express this class 2. We don't know short answer, but it's true that we identify them in several contexts, several biological contexts in our map, this class 2 signature. And it has been already shown by others in bibliography that some neutrophils express this class 2 molecules by flow cytometry, for example.
Do you think they actually present something on it, or?
It has been suggested, and there are some studies showing that, but we didn't approach this directly. The possibility is there, and I think there are evidences for this. We don't know how efficient they can present and if this is important for
Right.
adaptive immunity, but yeah.
Yeah. So we have another question. There have been reports for MSDC, sorry, that they can suppress by chemically modifying the TCR or by depleting local amino acids. Do you have any idea how the neutrophil mediated suppression is working?
Yeah, we don't know how this is happening. We at the beginning thought that there may be some soluble factors that they produce according to the transcriptome that may inhibit T cell responses, but we are now checking if this is dependent on direct contact with the T cell, for example. And so this is something we need to go deeper in detail.
So maybe a little related, there is a question on, did you test the suppressive effect of neutrophils on different T cells with different specificity? Does it impact different T cells differently?
Sorry, we just did these preliminary experiments in vitro with T cells that recognize OVA, and we want to move to a more physiological context using naive T cells experiments, but we are not experts on T cells, and so yeah, we don't. I don't know exactly how to do this. We need to find experts to do that.
Yeah. Another one is, do you have any clues about what could drive AP1 activation in lung neutrophils? Could the microbiome have a role?
That's a very interesting question. The microbiome in our previous study on neutrophils, specification in tissues, we took germ-free mice and we took neutrophils from tissues from these germ-free mice. And we were expecting to see changes in neutrophils in tissues in their transcriptome, but at least by bulk sequencing, they were identical. The neutrophils from a lung in a wild-type animal in a germ-free animal. So this kind of excluded the idea of the microbiome, at least for the lung, as a mediator of their specification. We don't know for other tissues, and we really believe that in the gut, there must be any contribution of the microbiome to neutrophils.
Interesting. And do you think neutrophils are primed during development in the bone marrow to later acquire one of the functional states you found, like immunosuppressive or interferon or angiogenesis or others?
Yeah, that's a future question to answer. I think this has to be performed using clonal tracing, for example, to make sure that the priming is happening in a specific populations. And this is something we are trying to develop. But the studies from Honbuluo lab, the studies on heterogeneity of neutrophils suggest that there may be some specific populations in the marrow that give rise specifically to neutrophils population in the circulation. This is a mystery indeed. I think it has not been proved that there may be specific clones that gives rise to specific neutrophils types. So yeah.
Very nice. I have another question. Are these neutrophil profiles maintained through a form of memory or trained immunity?
Yeah, that's also interesting. We don't know. Indeed, we don't know. We need to do the experiments. Yeah, but it's a possibility. Yeah.
Yeah, and you have an offer for a collaboration from a T-cell expert if you need some help with the T-cells. Another question is, did the CXCR4 KO mice do worse on DDC diet or better?
Yeah, that's something. Well, the CXCR4 KO mice, I'm going to, it's specific for neutrophils, okay? It's not the whole knockout is not available, but it's not alive. But that's something interesting. We know that the disease is different because in wild-type mice with diet, you have this expansion of the biliary tree, and the expansion of the biliary tree in clinics is associated with worse prognosis in the disease. It's a clinical symptom of worse prognosis. In the CXCR4, we have this parenchyma that is completely damaged. So the damage is in the parenchyma of the liver. We don't know what is worse. What we know is that it's a different disease, and that's what is pointing us to localization is important for disease outcome in this case.
Very interesting. I do have a question. You are showing a lot of organs, but one is never there. It's the brain, and actually, I'm working on the brain, and we have done some profiling on that, and we never see neutrophils, or at least I don't see them, and I don't know if there are. I'm just thinking, I never really pay attention, but now I'm thinking, are there neutrophils in the brain or not, or?
Yeah, so in the initial screening that was performed in the lab when I arrived to Andres Hidalgo lab, two tissues, the gonads and the brain, were the only tissues you could not find neutrophils in a steady state.
I see.
So we have neutrophils from the brain in our map, but after stroke. And we also have neutrophils from the meninges, which is not the brain, but it's close. I mean,
yeah. Okay.
Yeah, but
it's only in disease conditions.
Yeah, exactly. Yeah.
Very interesting. And did you look at sepsis, like septic condition? What type of neutrophils is going there in tissues when you have a septic shock or something systemic?
Yeah, we are including this septic shock right now in the map, but we have zymosan, for example. Zymosan induction, peritoneal neutrophils. Neutrophils, they acquire a very interferogenic signature. They move toward this interferogenic signature.
Yeah. Okay, that makes sense. All right. I don't see other questions in the chat, so I think we're probably good. Thank you very much for all the interesting discussion, and
thank you.
see you next time, everyone, for another seminar.